The genome of an artificial fungus was completed. Let me tell you about why this is so exciting. In case you missed it, the very first synthetic life was made back in 2012. That's Mycoplasma laboratorium. This involved designing and creating a synthetic genome. They had to create the nucleotides, and those can be made through inorganic means. I say inorganic, it has carbon in it, so it has to be organic, but it wasn't made in a life form. The first thing they had to do was create the nucleotides, and that can be done in abiotic ways. They assembled the nucleotides into genes, and those genes were assembled into a chromosome. In the case of a bacteria, it has a singular circular chromosome with just 10,000 base pairs on it. This research is in minimal genome work, and that's a field I work in too. They're trying to figure out the minimal genes required in order to create a functional cell. This has applications to figuring out how life started on Earth, and maybe what life would look like on another planet. We've also been able to incorporate non-canonical nucleotides, which I think is just really cool. So we have our adenine, thymine, cytosine, and guanine in our genomes, 4 base pairs, but there's many more that can exist. Some of them are made in our own bodies and are incorporated into proteins. But you can make them anything your heart desires, and you can assemble it into a genome of a microbe. They can even make that genome code for non-canonical amino acids and make proteins that could have never existed on this planet. But it tells us what life is possible on other planets. This does have a lot of applications. You can also make polymers, biopolymers, that could have never existed naturally. They might be stronger than existing polymers. They could be fabrics that are biodegradable that we haven't even thought up yet. But back to our friends, the yeast. Yeast are orders of magnitude more complicated than a bacteria. They have 16 chromosomes, and the final chromosome is 903,000 base pairs. In contrast to mycoplasma with 10,000, we're talking about hundreds and thousands. We have been making synthetic microbes for decades, but the idea of making a eukaryote is insane. When they assemble that genome, they have to consider what genes are on it that are going to support the mitochondria, which is like an entire bacteria on its own inside of the cell. They also incorporated a neochromosome that doesn't exist in nature into this little guy too. It was more than just figuring out what genes are necessary to live. That in itself is a monumental task. They had to figure out the right locations of those genes to interact with each other. Think about millions of base pairs, lines of code. Genes interact with each other in unexpected ways, and they kept trying, kept putting them at new locations and seeing if it would result in something functional until it finally did. The process of making these guys involves taking the husk of another cell, something that was alive, and replacing its genome. It's not that dissimilar to how cloning is done. That thing, if everything works out perfectly, will take advantage of its structures and come to life. This opens so many doors. I never thought that Blade Runner-esque organisms were possible, but now I'm starting to think, yeah, maybe we could do it. The next big step would be assembling an artificial animal. There are animals that are comparable to yeast in terms of the complexity of their genomes. We can do it. More importantly, the medical applications. Imagine somebody who has cancer. They went through treatment, radiation, and their genome has been damaged. We could actually replace damaged chromosomes. That's possible because we can assemble them now. That is, of course, a monumental leap from where we are now, but it's coming. The other part of this that I'm really excited for is the treatment of diseases. There's a lot of diseases that are just based on bad genes, bad luck. We're not that far from just being able to replace them. There's a lot of research right now in genetic therapies, and a couple of them have been cleared by the FDA for people. That includes a treatment for a kind of genetic blindness and a treatment for sickle cell anemia. Now, sickle cell anemia is a little bit easier to treat. That's because your blood is constantly made, so they replace the bone marrow with a functional set of genes. But being able to edit your own genes rather than needing a donor would mean a life without having to take immunosuppressants. I do hear a lot of people just scoff at this kind of work and say that it's not important or not incredible or it doesn't have returns. This unlocks a whole new world for research, things that we couldn't even conceive of studying. Suddenly, we can make these things. We are getting to the point where you could assemble a whole new organism to do your research on, design a gene, put it in a synthetic mouse. Yes, I know, that sounds kind of creepy. A lot of people like to ask me why I'm so excited for the Frankenstein-esque science. Or tell me that they're disappointed in me for being excited for it. I've always loved the spooky and the weird side of science. I've been able to create critters that have never before existed on Earth. I love the possibility of not nature-made creatures, but designed creatures. Is it a little bit spooky? Yes. When you work with this kind of stuff on the regular, it's a little bit less spooky. It's just really fucking cool.